kar malo prvi čujem. Hvala lepa za povabilo, seveda. Verjetno je, rad bi začel z zahvalo slovenskemu okolju, kjer sem se zelo veliko naučil na začetku. Zaradi tega bi šel rad skozi par imen z Inštituta Jožefa Stefana in univerze. Bi se rad zahvalil vsem, ki so mi na začetku pomagali, učiteljem v splošnem, ampak profesorju Turku še posebej, pa tudi Vidi Pizdar, Igorju Kregarju, profesorju Igorju Kregarju, profesorju Frančku Gubenšku, Brigiti Lenarčič, Metki Renko, tudi s Tamaro smo se križali na inštitutu. Na univerzi se zelo spominjam predavanj in vaj profesorja Stanovnika in tudi drugih profesorjev, tako da nič mi to ni nazaj držalo, obratno, zato sem zelo hvaležen. Zdaj bi pa prešaltal v angleščino, ker nažalost strokovnih izrazov v slovenščini bi bilo malo težko za mene na hitrico. Upam, da bo to v redu. The title emphasizes the word harmony, which may be a little bit strange for a scientific title, but I hope you will see that harmony is an appropriate term for different aspects of modeling in biology. First of all, models in science in general actually are computed by maximizing the harmony between the model and the input information, the experimental data on which the models are based. In a deeper sense, models once in hand also maximize harmony between the input data themselves. And finally, when models are interpreted in some sense, one maximizes the harmony with the rest of the knowledge about some domain and the model as well, through using the model for explanation of different facts. So I would like to break the lecture into three parts. The first one is quite a long one actually, will be a general introduction to integrative modeling in particular. I will define that for you, outline it. Then I'd like to illustrate integrative modeling by a few applications to specific biological problems in the domain of structural biology. And then I'd like to conclude by what we call meta-modeling, which is an instance of integrative modeling that I think is especially important for maximizing the impact of structural biology on cell biology in particular, where the systems that need to be modeled, like the cell, are even more complex than the systems that are typically of interest to structural biology, like individual proteins and protein complexes. And that's the area where we are combining models as opposed to data in order to compute a model of a system of interest. So a few general comments about modeling. There's many definitions of modeling, but clearly modeling is one half of science, which is a process of iterating through doing experiments, collecting data, and then interpreting the data in terms of models, generally speaking, so that we can understand our system of interest better and suggest better experiments or additional experiments in the future, and so the cycle repeats. A key purpose of a model is, of course, to be more helpful than the input information in rationalizing known information and so on. And models, model qualities that we would like to maximize, or at least optimize, are that it's better that it's computed in a quantitative fashion than described in words, for example. We'd like to maximize accuracy, precision, completeness of the model, because then obviously such models will be more useful for whatever applications we have in mind. And these terms, I'm pretty confident you know at least approximately what I mean by them, but they are visually depicted here. Accuracy and precision, completeness both in terms of extent and detail, and then these qualities will determine the explanatory power of a model in terms of connecting it with the rest of our knowledge and rationalizing and explaining things. To make two important points about modeling, I'd like to use a model that everyone knows, and that's the double helix model of DNA, proposed many years ago, more than 70 years ago, by Watson and Crick. This model was in fact inferred from multiple types of information about DNA. They knew about the X-ray, they had the X-ray diffraction pattern from Rosalind Franklin, which told them about the helical nature of DNA and also about the pitch of the helix. They also had the composition and stoichiometry information about the components of the DNA. They knew about the chemical complementarity. They were good enough chemists. And that allowed them to propose this model, the double helix model of DNA, which changed the world, as everyone knows. And this model makes two very important, simple, but I think important points. First, to understand and modulate cellular processes, we need their models, not only the data. And that these models are best generated, at least when they're difficult to obtain, they are best generated by considering all available information. No single piece of information on the slide is sufficient on its own, but in combination, they clearly allow them to propose this model. Structural biology is undergoing very rapid development, and maybe because of cryo-electron microscopy, computational advances, and so on, it's going through especially a rapid development these days. And it's developing in a number of different directions, but one of them is in terms of the size and complexity of the systems that are described structurally or spatiotemporally. And that's illustrated on this slide. In the 50s, the first atomic or near-atomic structures of proteins were determined by X-ray crystallography. We can now describe structures of systems that are 4 orders of magnitude larger, like the one in the middle here. And we're hoping to continue in this direction until in the end we will have some kind of a spatiotemporal description of systems as complex as the entire cell. That's another 4 or 5 orders of magnitude larger in terms of the number of atoms in it than the nuclear pore complex here in the middle. And our research group aims to contribute to this progress. It's clear that models, useful models, the best possible models of systems like this will require combining information from different sources. There will not be a single method, one expects, that will provide all the information required to obtain a satisfactory model of the cell, for example. So we need to combine information, just like in a smaller way, subjectively, Watson and Crick did to get a model of the DNA. And our group is correspondingly developing, applying, and disseminating such integrative modeling methods, both to facilitate basic science, but also biomedical discovery. And so we come to the definition of integrative modeling. It's just a type of modeling where you use different types of input information to compute a model. This information in general can come from different types of experiments, from different physical theories, from statistical analysis based on databases, and also possibly prior models. And obviously the motivation is that by using all available information, we will have maximized the accuracy, precision and completeness of the output models, if we can do it well. So that's the mindset that we adopt formally, right? Not ad hoc anymore. We'd like to develop formal methods for combining information of different types optimally. Before I tell you how we do that, I would like to increase your confidence in what you're listening to by showing you some sample applications of this approach that I have yet to outline to you, to different systems of different sizes, different complexities. These are structures in most cases, but also static structures. They are mixtures of structures. Sometimes they are models of dynamics of a system. Sometimes they're even models of networks that were produced by combining information from different sources. For us, always in collaboration with experimentalists who generate the data and help interpret the model. The first eukaryotic mammalian ribosome structure, the first nucleopore complex structure, the first spindle pole body structure, proteasome structure, for example, are some highlights. Okay, so now with this mindset, this is how we implement it. This is the workflow that we follow in order to compute models based on varied types of input information. We have this information in the input, and then we convert it into the output model in three ways. There are three end parts of modeling that depend on some subset of input information that are required to obtain the output models. The first one is what we call model representation, and that is just the set of all variables whose values are being determined by modeling. In structural biology, traditional structural biology, these are Cartesian coordinates of each atom in the protein. But at a lower resolution, we may be interested only in positions of proteins in a complex, only 6 degrees of freedom, position orientation, instead of a coordinate for each atom. And if we have a structure of a protein, of a subunit of a complex from prior experiments, we can use that information to model the whole protein as a rigid body to simplify the modeling problem later. So defining model representation is one important step in this modeling process. The second one is defining the scoring function, which is just a way to rank the alternative models based on the information available. You have to be able to say, oh, this is a better model, this is a worse model. A potential energy function is a special case of such a scoring function. NMR restraints are another example of such a restraint. A correlation coefficient between the shape of a model and the cryo-electron microscopy density map is another example of a term in a scoring function. The best scoring function, which I can't explain in any detail, is probably the so-called Bayesian posterior model density, which depends on what's called likelihoods and priors, each one of which reflects what the model is that you're assessing against the information, as well as the input, some subset of input information about it. And then the only other thing that we need is some way of searching among the alternative models, right? So once you know how to say which model is better, you have to be able to propose alternative models for this assessment by the scoring function. And so we need some kind of a search process, which invariably is a stochastic one, because the space of models is too large typically in structural biology, and so we use a version of Monte Carlo, one or another version of Monte Carlo methods here for the search process. And then we hopefully get some models, and hopefully we get models that need to be analyzed. And they are analyzed in various ways, but the two most important questions that one needs to ask are whether the models that you obtain satisfy both the information that you use to construct them, as well as information that you omitted from constructing the models. And if the answer is no to any one of these questions, you have to go back to the modeling process, maybe even to the lab to clarify experimental information or collect more of it. until you can say yes to this question. And then finally, you have to ask yourself, is the model or the set of models that you obtain sufficiently precise to have a chance of being useful for addressing the questions you're interested in? And again, if the answer is probably no, you have to get more information or change something else in the process until you can say yes to this and proceed with using the models. This is called statistical modeling approach, and we applied to integrative modeling mostly in structural biology. But as I pointed out to you already, this is a general approach. You can use it to compute different kinds of models, and they are illustrated here. Some are not even structural. This is a molecular network. The nodes are proteins. The edges are relationships between them. This is a trajectory. This is a free energy landscape. Can also be modeled using the same integrative modeling approach that maximizes the input information. And therefore, if you can do it well, also the output model's accuracy, precision, and completeness, which is what we want. So I will skip this in the interest of time, but I already registered that, in my opinion at least, the most objective and rigorous and appropriate scoring function is a Bayesian posterior model density. Some comments on it. I think I'd better move on. I would like, though, to relate the statistical modeling approach that I outlined to you to machine learning way of modeling, because machine learning is, of course, extremely popular these days and has some strengths and weaknesses. And it's good to relate the two to each other. In machine learning, one simple way of describing it is to say that the input is a training set that relates, for a set of cases, the input and the desired output. So a protein sequence and a protein structure that you wish to predict from sequence. Then machine learning will use a mathematical transformation, such as a neural network, and train its parameters in order to reproduce the desired outputs for each input in the training set. That's what machine learning does. And the trained neural network, for example, and it could be other transformations, will be called a machine learning model. It's a little clash between the terminologies, but that's how it is right now. And once you have a trained model, you can apply it to predict a structure for any protein sequence, in principle, to get a predicted structure for which you don't know, of a sequence for which you don't know a structure. So that's how machine learning works. Notice you need a training set, but which you don't for statistical modeling. But you don't need to know anything about the relationship between the input information and the modeled features that you do need to know in order to formulate statistical modeling. So you are in good shape if you have a training set, but it's a shortcoming for machine learning if you don't. The other point that I'd like to make here is that it seems to me that most people think in terms of statistical modeling, not in terms of machine learning, when they are explaining known facts or data about the system of interest with a model. So there's a certain transparency, a sense of understanding of what you're modeling when you're constructing a model with statistical modeling that you don't get, at least not yet, with most machine learning approaches at this point in time. And that may be important to you. Of course, one can aim, and we are working on this, as are others, on integrating the two approaches into a single unified approach that kind of minimizes the weaknesses of each and maximizes the strength of each of the two workflows that you see on this slide. Okay, so we are also developing open source, of course, software that implements all aspects of integrative modeling in our lab. It's a group effort, actually more than just our group's effort. It's kind of schematically indicated here. It's constructed modularly, so we can mix and match different kinds of system representations with different scoring functions dependent on different kinds of data, with different sampling schemes and so on. And we like to distribute it as broadly as we can and help others use it, and also develop some modules of it if they wish. And that's an advertisement. If anyone is interested, don't be shy, get in touch. Another way we try to maximize our impact is by contributing to the community through constructing a new module of the protein data bank, of the worldwide protein data bank, which is the single community archive, reference archive for structures, biomolecular structures. And so we contributed an archive that's developed for integrative structures in collaboration with the PDB. And in order to do so, we had to expand on three aspects of the traditional protein data bank. The first one was increase the richness of different ways of representing structural models. Traditional structural biology only specifies Cartesian coordinates of an atom, maybe one or two other parameters, but it's relatively simple in terms of the type of a description of a protein structure. Here we often need to use coarse graining, describe the structures at lower resolution. Maybe even in a multi-scale fashion, we need to combine, we need to describe a multiplicity of structural states in one sample. We need to order different states with respect to each other as well. So we needed to expand on that representation in this fashion. We also constructed the archive with the structural module in the middle, but connected in a federation of archives with independently or semi-independently constructed archive that contain data of different kinds, many more kinds than traditionally used in structural biology, which is crystallography, electron microscopy, NMR spectroscopy. But in integrative modeling, there's many other kinds of data that are used for computing integrative structural models, and we needed to archive them as well as the models themselves. And in order to make it scalable in the future, when we hope the resource is expanding, adding new experimental modalities to the family, so to speak, so we can increasingly cover cell biology and not just structural biology with this approach. We had to make it into a federation. And then finally, we have to pay a lot of attention, maybe more, to assessing the models and data than to modeling itself, because obviously one needs to know the uncertainties involved when you're interpreting the model. And we have an ongoing effort that's grounded in Bayesian inference and will be general across different types of structural models in the PDB on this front. Okay, so that was my introduction. And now I will illustrate it briefly, integrative modeling, that is, by outlining two applications, I think. And we'll be focusing on the nuclear pore complex and the NPC, which is a large protein complex. It's about 50 megadaltons in size in yeast. It consists of 500 or so different proteins, which are protein copies, subunits, which are called NUPs. And they are of 30 different types. So you can see an 8-fold symmetry in these images already. There's a symmetry in the system. That's why there's only 30 different types. But in total, there's 500 proteins coming together. It's important in biology because it regulates transport of proteins and RNA between the nucleus and the cytoplasm. It sits in the nuclear envelope, which is colored in green, and the complex, the NPC, is this blue ring. The transport occurs through this hole in the middle of this ring. And we were interested in the structure of this system in order to say something interesting about these other aspects of the NPC, including evolution, function, mechanism of assembly, interactions with other systems in its neighborhood. All of this obviously, potentially at least, leading to our ability to both modulate the system for studying basic biology, but also perhaps for therapeutical purposes. And from the beginning, this has been an effort with absolutely fabulous experimentalists, Mike Rowett and Brian Chait, but also others. And I pointed out that everything we do with the real systems is in collaboration with experimentalists, and so was the case in this project. So let me tell you about how we determine the structure, or outline how we determine the structure, and I'll pick this study from 2018 to tell you this story very briefly. And first I'll say that this slide is just another depiction of the same workflow that I already shared with you. So I hope you will recognize that it's just in a different format. So what's the first step then is collecting or gathering the input information. What is the input information? The main types, at least, that we used in this case. First and foremost, we had information about composition and stoichiometry from different versions of mass spectrometry, some of them pretty fancy, but it was important to know what are the components of the system, including their copy numbers, of course, before you can model its structure. We also had information about the shapes of many of these components, and sometimes even their smaller complexes, say 2 components, from prior structural biology studies, including various methods as indicated here. And now the problem became, how do we assemble these rigid components, or almost rigid components, together, 500 plus of them, of 30 different types, into this ring-like structure that you saw on the previous slide. And that information came mostly from 2 sources, experimental sources. First of all, we had over 3,000 distances between pairs of lysine residues, mostly, in these proteins. And so if you imagine in a nuclear pore complex isolated from yeast, in hopefully native state, more or less, you expose it to a chemical crosslink that's at most 20 angstroms across, has 2 functional groups on each side that interact with the lysine residues. So only those pairs of lysine residues that are closer than 20 angstroms will be detected by subsequent proteolysis and mass spectrometry as being cross-linked to with this reagent. And that's how we determine that distance restraint. You have 3,000 of them, not very precise, each one of them, but you have over 3,000 of them. And then we also had the approximate shape of the whole system from a version of cryo-electron tomography, where I'm not explaining what that is, but I'm assuming most of you are aware, with a certain amount of averaging of multiple particles and considering the symmetry to obtain a map that's, at least in 2018, was at a decent resolution of about 25 angstroms or so for much of this ring. So then what we had to do is start with a totally random, if you look at this movie here, at some point it'll start here with a totally random constellation of components. We don't know how they're assembled, we just position them randomly. And then we run a Monte Carlo search process that moves the positions and orientations of these components randomly in millions of steps, each time assessing a slightly new configuration against the input information. Does it reproduce the distances observed experimentally? Does it have the shape observed experimentally? Does it have the symmetry? Do the membrane proteins span the membrane, which is part of the model here? And over millions of steps, hundreds of thousands of independent calculations of this sort, we will find, in this case, a set of models that do satisfy the input information sufficiently, and this small cluster of them is shown here in the bottom. That's the result. So we just follow the workflow I introduced to you for this specific case. I hope you have a little bit better sense about how it works, integrative modeling. The end result is a set of structures, each one of which is as good as any other, or structural models, I should say, is, that have about a 9 angstrom RMSD, root mean square deviation between them, and we can define that as the precision or equivalently uncertainty of our model, making some other assumptions about it, but roughly that's our quantification of model uncertainty, and that's how the model looks. In gray, you have a nuclear envelope, a cross-section or small section of it. In brown, there's a so-called membrane ring consisting of proteins that anchor the NPC into the nuclear envelope, on top of which membrane ring, there's inner rings, and then there's outer rings on top of the inner rings, and then there's this functionally important modules of the system that interact with cargo as it passes through. And maybe most important functionally, there's the FG naps on the inner side of the pore. I know it's difficult to see here, but these green spots are globular domains of the FG naps, called FG repeat anchors, that line up the inner side of the pore, and then the sorted regions called FG repeats, or these spaghetti-like lines, fill up the pore, and it's thus FG repeats that allow the system to regulate transport between the nucleus and the cytoplasm, and that's a separate story, but and that's how the structure looks as a whole. Clearly, one needed to do integrative modeling in order to get to this point. None of the individual pieces of data would be sufficient. I'll just say that this illustrates very nicely and concretely how, by increasing the amount of input information, one increases the precision of localizing a specific protein. Initially, you don't know much, or basically nothing, and as you add more information here of different types, you can localize the component protein more and more precisely. So it's kind of a nice, I think, visualization of how integrative modeling makes sense. Right? You need to combine all of this in order to get to this localized spots for a protein, as opposed to only knowing that a protein is somewhere in the cylinder, but not where. So I showed you one. I guess I showed you this one. But in reality, there was a series of progressions starting over time in terms of how complete and precise the models of the NPC were. And initially we had just a model of topology shown here, and by adding more and more information, improving modeling, over the years we came up with a basically near atomic structure now of the entire system, even including the so-called nuclear basket, which in this case is on one side of the NPC. So that was a few words about integrative structure characterization of the NPC. And now I'd like to tell you how we modeled recently the process, not a static structure, but the process of how these 500 plus proteins come together in the context of the nuclear envelope to assemble a mature nuclear pore complex structure. And that was a collaboration with Jan Ellenberg and his postdoc Shutura, who was at the time at EMBL in Heidelberg. And this I'll go through even more shortly, but just to give you a sense. The nuclear pore complex assembly is obviously an important process for eukaryotes. This is a cell cycle of a typical eukaryotic cell. You cannot have a hole in the nuclear envelope that's not regulated. That would not be a viable cell. You also can have a nucleus without a hole regulated by the NPC. So the assembly and disassembly of the NPC during the cell cycle is an important process. It was important in the evolution of eukaryotes, and so we study it. It occurs twice in a typical eukaryotic cell along the cell cycle in different ways. Actually, the mechanisms are different. Here is a schematic picture of one of these two types of NPC assembly that was based on a lot of genetics and microscopy and biochemistry, kind of put together manually or subjectively by smart scientists who know all the papers and so on. And so that was our starting point. Our goal was to improve on this by formal integrative modeling as much as we can. And this is what we did. So again, the same format as before. This is the main types of input information, and then I'll tell you how it was converted in the model. But the main types of input information, first and foremost, was the mature NPC architecture that I just described to you. Obviously, any assembly has to end in the mature assembled NPC structure. I haven't emphasized the physical principles before, but obviously, we considered things like excluded volume and so on here as well. Proteins can't overlap in space, so there's a few other physical principles I'm not mentioning, but it's an outline. And then very importantly, there were two major temporal data sets. The first one was information about about the composition and stoichiometry of the growing intermediates of the NPC. The process takes on the order of 20 minutes, so it was possible to do time-dependent fluorescence correlation spectroscopy, which relies on tagging NUP components in order to count how many NUP molecules occur in the growing assembling NPC at different points in time. So on the order of 6 snapshots could be characterized that way. And then fortunately, at the same time points, it's also possible to take a snapshot of the shape of that growing nascent complex using a single particle time-dependent cryo-electron tomography. So we had the approximate composition, including copy numbers, and approximate shape as a function of time. And then what we did is we first imagined that the assembly process occurs from left to right in this schematic, where you have an alternative structural states at each time point. This is at the beginning, this is at whatever, 5 minutes, 8 minutes, and so on, moving to the right. At each time point, there are alternative structural states that are informed by this experimental information, physical principles, and the need to begin resembling the mature NPC architecture, the more so as you move from left to right. But there will be alternatives. And then we needed to connect these alternatives across neighboring time points. So there's multiple structural states at each time point, and maybe thousands of them. And then those that make most sense are those that allow for a facile transition from one time point to another. Right? You don't want to completely unravel a structure at 5 minutes in order to get to 6 minutes. A more reasonable transition between 5 and 6 minutes among the alternative states at 5 and 6 minutes is preferred as you score a model of the process. And in the end, if you go through this, you actually get a single, at some resolution, you get a single assembly pathway described in this discrete fashion that is much more explanatory of the input information than the alternatives. So again, using those assessment considerations I outlined to you before, we declare relative success and move on and interpret this model in various ways. But I'm not going to describe that. I'll just telling you how we computed it today. So again, we have different types of input information. We computed, in this case, a spatial temporal model, not a static structure model. And now the last few slides, I know time's running out. I would like to begin introducing the idea of the need to combine different types of models into a bigger model, as opposed to combining different types of data into an integrative model. And I think that's an important concept for modeling the most complex of the systems in biology, because it divides and conquers the bigger modeling problem into a set of smaller modeling problems whose solutions are then combined together to solve the big modeling problem. And in fact, it's illustrated quite nicely even with the nuclear pore complex system here. Over the years, we we contributed a number of different models, different types of models of different aspects of the MPC. I talked about the static structure model. I talked about the assembly pathway model. But we also described, we proposed how the MPC might have evolved, how it functions, how it regulates transport through the pore, and then how it interacts with the cell-wide so-called RAN GTP network that provides the energy for the transport to be irreversible as opposed to reversible, which is what the model here represents. And then finally, we also are mapping the interactions between the NPC and the neighboring systems that are, of course, transient and heterogeneous. These neighboring systems will include things like chromatin, spindle pole body, proteasome, ribosomes. These are other things in the cell around the NPC that are coming, that are transiently interacting with the NPC and each other in some cases, and we are expanding the scope from describing structurally a single protein complex to describing a neighborhood around the target complex. And so these are just different types of models, and clearly in order to maximize our understanding of the NPC, we wish to combine these models with each other. We wish to formally, automatically, and optimally combine these models with each other and not compute them independently as we did for those panels here. And that we call the meta-modeling problem, and we proposed a version of meta-modeling which we call Bayesian meta-modeling that we illustrated with a simple toy case, but it's very much still this sort of exploration work in progress. But let me just belabor the point a little bit more, because I do find it very exciting, plus it's the very much current topic of our interest. This just makes a distinction between integrative modeling, as I described it so far, and meta-modeling clearer by providing another example. See, in integrative modeling, you can imagine having cryo-electron microscopy data, maybe a cryo-EM three-dimensional map of some system like the ribosome, and some chemical crosslinks from mass spectrometry, different kinds of data that are integrated, combined together into a structural model like this, just like we did it for the MPC. But in meta-modeling, what you'd like to do, for example, is combine a structural model of the ribosome, the same one as from the previous panel. You can like to combine it with some kind of a molecular network model where these nodes are proteins and the edges are some sort of an interaction between them. Doesn't have to be physical, could be functional, doesn't matter for this example. You'd like to combine these two types of a model to get a more complete picture about how the ribosome works in the cell. So that's the contrast, and my suggestion, hypothesis, opinion is that in order for structural biology to contribute to cell biology, we would benefit if we can, from meta-modeling, combining very much different types of models of different aspects of different parts of the cell, from network models, structural models, spatiotemporal models, compartment models, diffusion models. All of these are different types of models that are computed usually by different people based on different types of input information with different modeling methods independently, and yet they're all about the same cell in principle, or the cell. And so can we put it all together into the biggest picture we can? And that's the goal for meta-modeling. It divides and conquers modeling of really complex systems, and therefore I think it's worth pursuing. I know time is an issue, so let me rush through a simple toy model where we demonstrated the idea. We were concerned with glucose-stimulated insulin secretion in pancreatic beta cells, which is clearly a multi-scale system. There's organs like liver, pancreas, obviously. There's cells. There's pathways in cells. There's molecules in pathways, small molecules, biomolecules. And there were a number of different types of models that describe different aspects of this hierarchy, including things like ordinary differential equations describing signaling, some signaling processes, metabolomics models. This is a model of dynamics. I don't know why it doesn't move, but it's a Brownian dynamic simulation of diffusion of insulin granules inside the cytoplasm that precedes the excretion of insulin from the pancreatic beta cell. And there's a structural model of how GLP-1 receptor interacts with GLP-1 hormone, but could be replaced by agonists or antagonists. So there's a little bit of structural biology here and so on. And how do you combine these models together? Well, first, all these different types of models are converted into a unified description that relies on what's called probabilistic graphical models in computer science that conveys the statistical distribution of variable values and their statistical independencies. And I'm not describing how we did this. Obviously, I'm giving you just a very coarse outline. Once each one of these very different types of models are converted into this common language, mathematical language of probabilistic graphical models here, we define what we call couplers between the independent models. That is just another set of models that share at least one variable from one model and at least another variable from another model, thereby coupling the two models, making them at least statistically, if not quote-unquote physically, interdependent. And with those couplers in hand, then the only other thing that's left to do is what's called backpropagation, commonly, where you update the distributions of all the variables in your probabilistic graphical models to make them more statistically consistent with each other. And these updated models are the output, collectively, a meta model. That's the approach. If you have good physics about some connections between some of the input models, you can input it within this framework as well, if you have it. But if not, at least you have this more statistical approach to still couple the models with each other and still hopefully get some benefit from considering much more input information in meta-modeling when you're combining input models with each other than you would when you're considering each model on its own. There's some advantages in principle of this kind of modeling. Only one of them is illustrated here, which I find particularly exciting because it's all about maximizing the understanding of the system that's modeled. Imagine that in the input, business as usual, you have two separate models that include but the variables x and y respectively. You can't say anything about the relationship between x and y when they occur only in the two separate models. They have nothing to do with each other. But through the construction of the meta model, you couple the two variables to each other, and now you can see whether changing the value of y changes x and vice versa, and how. And you can look for strongest dependencies of this kind once you have a meta model in order to increase your understanding of the system. And one specific non-trivial example is this nonlinear relationship between various concentrations of glucose insulin as a function of time that's called the incretin effect experimentally, that a meta model of ours will reproduce, and since you now have a model, at least in some level rationalized. And I will just briefly maybe repeat 6 points. I don't know if they're the best ones to repeat, but it's a selection for the presentation and then conclude here. Integrative modeling is a general approach to modeling based on different types of input information. Importantly, allowing you to maximize the input information. We do develop a software package called IMP to facilitate integrative modeling, especially for structural biology, but it's a general approach. We have helped by contributing a module of the protein data bank that archives integrative structure models and data, and in the process hopefully increasing the traction of integrative modeling a little bit. We developed and applied it to spatiotemporal modeling, not just modeling of static structures. We illustrated it with modeling the post-mitotic assembly process of the human nuclear pore complex, And then I introduced the idea to you of, or maybe not, but I talked about the idea of combining different types of models as opposed to different types of data, which we call meta-modeling. And I believe will be important for modeling the more complex of the biological systems. And finally, it's always in integrative modeling that you require a lot of contributors, because you're putting different things together and no one can cover everything. So it's difficult to explain it in a short amount of time because you have to talk about 10 different things, and it's difficult to pull it off because you need 10 different people and so on. And so the acknowledgements also are long. On our side, the main postdocs in our group at the time who worked on the problems I mentioned, the specific NPC problems, are Andrew, Jeremy, and Barak. They are all on their own. Andrew is at NASA, is not indicated here. There were others over the years who contributed to the NPC who are listed here. Jan Ellenberg, Mike Rout, Brian Chait, the key experimentalist collaborators. The whole leadership of the Worldwide Protein Data Bank is obviously supportive and helpful in our PDB contributions. Meta-modeling actually started by the challenge of modeling the pancreatic beta cell in the context of this consortium, which doesn't actually exist anymore, but we wouldn't be where we are without it initially, and some other funding things and so on. Thank you for your time, patience, and attention. Thank you.